Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Ellen Capon, Editorial Assistant at Drug Target Review. Today's speaker will be Luke Osteich, Associate Study Director, Lead Hunter Services, California, Eurofins Discovery. Following his presentation, we will move on to a live question and answer session where you can pose questions to Luke. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to Luke.
Everyone, and welcome to our webinar. We're glad you could join us. My name is Luke Osteich, and I'm an Associate Study Director with Eurofins Discovery. Today, I'm going to be talking to you about functional cell-based activity profiling of multi-receptor anti-obesity therapies. Our agenda for the webinar starts with a brief introduction to Eurofins Discovery services. I'll then give some background on related pathologies and molecular basis of obesity and the current state of anti-obesity therapies in clinic and on the market today, before moving into our case study, where we'll present a sample workflow for multi-receptor anti-obesity therapies using PathHunter cell-based assays, which will include data from market-leading therapies. Eurofins Discovery is your end-to-end provider for metabolic disease services. Today, we'll be focusing specifically on obesity, but we can support a variety of metabolic programs for diabetes and other therapeutic areas as well. We have significant chemistry and biotherapeutic capabilities and can work with a range of modalities. We have thousands of ready-to-use primary and orthogonal in vitro pharmacology assays, a subset of which will be the focus of our talk today. We have translational cellular assays to support your metabolic disease programs, which include biomap and custom assay capabilities using relevant primary cells and patient-derived materials. And finally, we support both in vitro and in vivo safety pharmacology with animal models of various metabolic diseases. Obesity is a multifaceted and diverse condition that affects more than just a person's weight. There are a multitude of health consequences that are associated with obesity that can impact nearly every part of the affected individual. As you can see on the slide here, these include both things that are physical, like cancers, heart attack, stroke, diabetes, among others, as well as mental, such as increased depression and anxiety. According to a recent study, greater than 14% of the world population is obese, and this number is expected to reach 25% by 2035. The economic burden that's associated with this epidemic is in the trillions of dollars. Relatively recent advances have revealed many of the molecular mechanisms between the gut and brain that control appetite and eating behavior. Various gut-brain peptide hormones are secreted in the gut, as you can see in the diagram here, in response to nutrients and other factors, and these include leptin, insulin, glucagon, amylin, peptide YY, glucagon-like peptide 1, gastric inhibitory polypeptide, orexin, and others that then travel through the circulatory system to the brain to signal through their cognate receptors. These are primary G protein coupled receptors, and they elicit additional signaling through neurotransmitters such as melanocortin and neuropeptide Y that ultimately control the body's appetite and caloric intake. The pursuit of weight loss drugs has been extremely challenging, and prospective therapies have ultimately failed or seen reduced use even after approval for a variety of reasons, which include adverse cardiovascular effects, increases in suicide risk, and dependence and abuse. As the molecular mechanisms that control appetite showed on the last slide became more well understood, new therapies emerged that target the glucagon-like peptide 1 receptor, GLP-1R. Now, this started with the approval of liraglutide in 2014 and really accelerated in the last 4 years with the approval of semaglutide in 2021. Semaglutide was the first drug to demonstrate consistent double-digit reductions in body weight in clinical trials. And since this approval, multiple additional GLP-1 agonists of different modalities have entered the clinic, with many targeting additional hormone receptors, including the gastric inhibitory polypeptide, glucagon, and amylin. As you can see from the timeline here, there have been more than a dozen trials reporting data already in 2024 for these different multi-receptor therapies. And more trials are reporting all the time, with many of them investigating either daily oral administration and weekly or monthly injectables. For today's talk, we're going to present data from 3 of these market-leading anti-obesity therapies, and I'll give a bit more details on each of them before we start. The first I mentioned on the last slide is Semaglutide. Semaglutide is an approved GLP-1R agonist that's based on the endogenous GLP-1. It contains a modified lysine residue that is lipidated and an alpha amino isobutyric acid residue, and both of these help to prolong the half-life and increase stability of the drug in the body. Semaglutide has been approved since 2017 under the commercial names of Ozempic and Rybelsus for diabetes, and it's been approved since 2021 with the commercial name Wegovy for obesity. In a phase 3 clinical trial for obesity, semaglutide administration resulted in weight loss of about 15% at 68 weeks. The second drug, Tirzepatide, is from Eli Lilly, and it's a dual GLP-1R GIPR agonist. If you look at the sequences down at the bottom, this is a modified GIPR peptide that's engineered to have additional GLP-1R activity. It also has these additional modifications that promote stability in the body. In a phase 3 trial, it showed weight loss of about 15 to 21% at 72 weeks, which is an increase compared to semaglutide. It's been approved for diabetes since 2022 under the commercial name Mounjaro, and since 2023 under the commercial name Zepbound. Finally, we have Retatrutide. This is a peptide that is also from Eli Lilly, and it is now a triple GLP-1, GIP, and GCG receptor agonist. It's based on the endogenous GIP with those additional substitutions that impart both GLP-1R and GCGR agonism. This drug is currently still in clinical trials, but in phase 2 clinical trials, it showed even more increased weight loss with about 17% at 24 weeks and almost 25% at 48 weeks. And now it is currently undergoing multiple phase 3 trials for both diabetes and obesity. Today, we'll be using these 3 drugs in a case study to show a sample workflow that can be used for profiling multi-receptor anti-obesity therapies using cell-based assays that I'm going to lay out for you next. Before I begin, I do want to note that this is really an example of a potential workflow, and the order of these steps that we're describing could be altered to fit the specific needs of any given discovery program that you're working on. Our workflow that we're presenting starts with target validation or focused hit finding using the obesityLITE panel of cell-based assays. It proceeds to potency determination in multiple formats for active targets, followed by broad specificity testing with the gpcrMAX panel, and wraps up the discovery and development portion with in-depth mechanism of action studies using gpcrBIAS, internalization, and orthologous assays. Finally, we'll present a little bit of preclinical validation data with safety pharmacology, and this step can also include pharmacokinetics and pharmacodynamics and in vivo testing. I'll start by giving a quick background of the technology that we use in our cell-based assays, which are PathHunter cell-based assays, and the technology is enzyme fragment complementation, or EFC. EFC technology consists of a split beta-galactosidase enzyme that features 2 components, an enzyme donor and an enzyme acceptor. These fragments are inactive separately, but when brought together in close proximity, will complement to form an active beta-galactosidase enzyme. The 2 fragments can be tagged to proteins of interest to interrogate biological events, with addition of luminescent substrate, resulting in generation of light that can be measured on a plate reader. This technology has been applied to a variety of different cellular targets, including GPCRs, G protein coupled receptors, kinases, interleukins, nuclear hormone receptors, and epigenetic and other pathway assays. These assays are suitable for use with a variety of different modalities, which include small molecules, peptides, large biologics, antibodies, or antibody drug conjugates. For the first step in our sample workflow, I'm going to introduce the obesityLITE panel of cell-based assays. If you recall from the introduction to gut-brain appetite signaling from a few slides ago, where I showed this diagram, there's a variety of signal peptides that are used, and the majority of these receptors involved are GPCRs. We took this signaling axis and assembled a fit-for-purpose collection of cell-based assays we call the obesityLITE panel. If you take a look on the right side of the screen, you can see the targets of interest in the GPCR or in the obesityLITE panel, which consists of 25 assays that measure activity against 13 receptors from the glucagon, amylin, cholecystokinin, ghrelin, leptin, melanocortin, orexin, and peptide YY families. Each receptor is profiled in the assay mode, either agonist or antagonist, that is most relevant for anti-obesity therapies, and the panel includes both beta-arrestin and second messenger assays for GPCRs. I'll go into more detail about these assays in a few slides, but first I want to present data for the 3 anti-obesity peptides we have already discussed. In the center of the slide now is obesityLITE profiling of semaglutide, tirzepatide, and retatrutide at 1 micromolar. The data is plotted as a response efficacy relative to the maximal signal from the reference agonist for each receptor on the y-axis. I'll note here that we're showing a single concentration for clarity on the slide, but the panel is run as a full dose response curve that gives potency measurements directly. On the x-axis, we have the targets tested in the panel, which are split into 2 groups of assays, either beta-arrestin or second messenger format. We'll talk about those in the next slide, but first I'll direct your attention to the data here. We have 2 boxes that are outlining the beta-arrestin and second messenger responses of our 3 peptides in our targets of interest. GLP-1R on the top shows activity for semaglutide, tirzepatide, and retatrutide. For GIPR, we have expected activity at tirzepatide and retatrutide. And for GCGR, we only see activity against retatrutide. I want to point out there's this variation in efficacy between the beta-arrestin signaling on the left and second messenger formats on the right, which is some interesting pharmacology we'll discuss a bit later in the talk. Before that, I'm going to give some details about what exactly these 2 assay formats are. GPCRs, as their name described, signal through G proteins and eventually recruit beta-arrestin. If you take a look now on the bottom left of the slide, we utilize EFC technology to measure beta-arrestin recruitment. In this assay format, both components are tagged, so it's a highly specific measurement with a direct 1-to-1 stoichiometry that makes it very good at identifying partial agonists. We have data at the bottom for the melanocortin 4 receptor, where you can see a dose response curve for the reference agonist, melanocortin 2, and no response from our 3 anti-obesity peptides. The second assay type in the panel is second messenger signaling. GPCRs are coupled to a variety of G-alpha proteins to affect downstream second messenger responses. One of these is modulation of cyclic AMP by GI and GS coupled receptors via adenylate cyclase. Now, we utilize our EFC-based assay in a competition format, which measures intercellular CAMP either increases or decreases. Because of downstream implication of second messenger response in these assays, they're typically more sensitive than beta-arrestin. You can see here again that we have data for MC4R, which is a GS-coupled receptor, so it increases CAMP levels in the cell. The 3 peptides, again, are not active in this assay. The other second messenger signaling that we can measure in this panel is calcium mobilization. Other GPCRs are coupled through GQ, which signals through phospholipase C to release calcium in the cell. We utilize a calcium-sensitive dye and a flipper to measure this release in real time, as opposed to our EFC format. The cholecystokinin receptor, CCK-AR, is a GQ-coupled receptor, and again, we have dose-response curve data for the reference agonist, CCK8, and no response from our 3 peptides. The final target in the obesityLITE panel is the leptin receptor. This is actually not a GPCR, but is a cytokine receptor. cytokines dimerize upon activation with their reference agonist. And so we use the EFC technology in this assay by putting a tag on 2 different copies of the receptor to measure activation after this dimerization occurs, the leptin response curve and inactivity of the anti-obesity peptides is shown again at the bottom of the slide. Now, overall, the ObesityLITE panel enables target validation or hit finding against a focused set of receptors that are important for controlling obesity in one single package. From this panel, we can take hits and verify on-target activity in both assay formats in the next step of our sample workflow. Here, we're now showing dose response curve testing for Semaglutide, Tirzepatide, and Retatrutide against GLP-1R, GIPR, and GCGR in both beta-arrestin and CAMP formats. Each graph on the slide additionally shows reference response curve data with extended 4 for GLP-1R, GIP for GIPR, and glucagon for GCGR. Starting with our GLP-1R beta-arrestin on the top left graph, you can see that each of the 3 peptides are active with potencies in the nanomolar range. Differences in partial agonism are clear, with semaglutide showing full agonism, retatrutide more partial agonism, and tirzepatide being minimally active. This is in contrast with GLP-1R CAMP on the bottom left now, where each of the compounds are full agonists and now have potencies in the picomolar range. If we move to GIP in the center, we have GPR beta-arrestin recruitment on the top. We now have no activity for semaglutide and have full or partial agonism for our other 2 peptides, again in the nanomolar range. Below that, we have our cyclic AMP measurement for GIP, where we have full activity of tirzepatide and retatrutide, and again, much more potent responses in the picomolar range. Finally, on the right side here, we have glucagon receptor data. We now only have activity for retatrutide, and it is either partial and nanomolar potency for beta-arrestin or full and picomolar potency for our CAMP measurement. This pair of beta-arrestin recruitment and CAMP signaling assays works great for use in structure-activity relationship studies to develop potent active molecules, as they can measure compound responses from really in the micromolar range down to the low picomolar range, as we see here. Now that we have confirmed the potency for active targets from the ObesityLITE panel, we can additionally confirm specificity against a larger selection of GPCRs in the gpcrMAX panel in the next step of our sample workflow. The gpcrMAX panel includes 168 Class A and Class B GPCRs that are assayed for beta-arrestin recruitment in both agonist and antagonist modes. This is typically done at a single concentration, but multiple concentrations can be tested if desired. On the slide here, we have phylogenetic trees of all the receptors included in the panel. Surrounding this on the left is agonist data, and on the right is antagonist data for our 3 peptides of interest. There are dots shown with the size of the dot reflecting the % efficacy of the response. I'll direct your attention now to our zoomed in insert that is just looking at the Class B GPCRs, which include our 3 targets of interest, GCGR, GIPR, and GLP-1R. We have data here for our 3 peptides, which show the responses that match up with the beta-arrestin data that we showed in the previous slide. I do want to point out here that we have very minimal off-target effects. These compounds are very specific, and we can see with our tree here that even in the most closely related Class B GPCRs, we have very minimal off-target effects. Overall, the gpcrMAX panel is a great tool for confirming compound specificity and identifying unwanted off-target effects, but it can also be used for target ID at an earlier stage in a program as well. Now, before I move on to the in-depth mechanism of action studies using gpcrBIAS, internalization, and orthologous assays, I want to introduce the principle of biased GPCR signaling. As we've already shown, GPCRs signal through both beta-arrestin and G protein pathways to affect their responses. And the principle of GPCR bias states that certain ligands can preferentially elicit a response from one pathway versus the other. while the biased agonist that you can see in the center of the slide equally activates both beta-arrestin and second messenger pathways. On the left, we have a beta-arrestin bias ligand that preferentially signals through only the beta-arrestin pathway and minimizes that G protein signaling. On the right, we have a G protein bias ligand that signals through G protein signaling pathways as opposed to beta-arrestin. And in both these cases, different downstream responses are going to be affected. Beta-arrestin typically has internalization of the receptor and can lead to desensitization. And a lot of times the G protein signaling is the beneficial pathway for responses that we'd like to see. This signaling bias towards G proteins and away from beta-arrestin has been proposed and demonstrated to be therapeutically beneficial for a handful of different receptor classes, which includes opioids, and specifically for GLP-1R and some of the other obesity-related receptors, such as melanocortin-4 and ghrelin, that we've talked about in the obesity panel. Various publications have indicated a benefit of bias signaling. This includes anti-diabetic effects and protections against obesity, which make bias an intriguing prospect for potential therapies. For GLP-1R, if you now look on the right side of the slide, second messenger pathways are believed to be responsible for appetite suppression, prior protective effects, and insulin secretion, while signaling through beta-arrestin can lead to desensitization that attenuates some of the beneficial activities that you see from activating the receptor. Because of differences between beta-arrestin and second messenger assays that we've already discussed in this talk, comparison of potency measurements between the two cannot be used to directly evaluate bias. Dr. Tirzepatide has published a model that allows for comparing responses between different pathways by deriving a transduction coefficient that utilizes ligand and pathway-specific parameter tau and the ligand receptor-specific parameter Ka. I don't have time in this talk to go into detail about the underlying mathematics, but these parameters can be used to calculate a log bias value that compares responses between two pathways for a compound relative to an unbiased reference compound. On the left side of the slide here, we have data for exenum-4, which is our neutral reference, and compared it to Semaglutide, Tirzepatide, and Retatrutide for GLP-1R beta-arrestin, which is the dotted line, and CAMP signaling, which is the normal line. The data is plotted slightly different than previous data we've shown because bias calculations require normalization to the maximum possible response in a given assay system. For this, we use forced limb stimulation in our CAMP format, and we use an EFC control in our arrestin format, which allows us to determine this maximum possible signal in each assay, which would be a 1 on our relative activity axis there on the y-axis. I'll move now to the graph on the right, which really shows calculated potency biases on the x-axis and relative intrinsic activity, which is related to efficacy on the y-axis. On this plot, values greater than 0.5 are generally considered to be significant, and this data shows that we measure slight but minimal bias for the three compounds, but actually a strong efficacy imbalance for Tirzepatide, which is reflective of the minimal efficacy that it shows in the arrestin assay. This is generally in agreement with published data from Eli Lilly for Tirzepatide, which has demonstrated that it is an imbalanced agonist. To delve deeper into signaling intricacies of each peptide, we also measured bias relative to arrestin in additional signaling pathways. So I'm going to start here with the diagram and plot in the center, which is GLP-1R activated internalization. This format measures beta-arrestin dependent receptor internalization. It uses our EFC complementation system again. Our EFC fragments are tagged to beta-arrestin, and another one is tagged to the endosome in the cell. If you look at the chart on top, you can see the response for each compound is similar to the beta-arrestin data we've previously shown. We can readily see partial agonism, and we have nanomolar potencies measured. Moving just to the right now, we have our total internalization format. This measures all forms of internalization as the GPCR is directly tagged along with the endosome. Interestingly, in the chart above, we now have more full agonism for each of the 3 peptides tested in comparison with exentin-4. And this suggests beta-arrestin independent forms of internalization may play more of a role for Tirzepatide or Retatrutide in this assay. Finally, on the right, we have beta-arrestin-1 signaling. Now, this assay is identical to the beta-arrestin assays that we've shown so far, but it now utilizes a different isoform of beta-arrestin 1 instead of beta-arrestin 2. The responses in this assay are very similar to what we've seen with beta-arrestin 2 so far. I'll now point your attention over to the left side of this slide, where we have our quantified signaling bias versus beta-arrestin 2 for the CAMP data we showed in the previous slide, and now our active and total internalization and beta-arrestin 1. On the bottom, we have our relative intrinsic activity imbalance, which was on the y-axis on the previous slide for the same 4 comparisons. We did not detect any significant signaling bias in this comparison with beta-arrestin 2 in these assays, but again, we do see that efficacy imbalance with Tirzepatide in the CAMP assay. Overall, this platform supports targeted development of compounds that have very specific pharmacological properties. Measurements of bias are not limited just to GLP-1R, but we can also interrogate other receptors that are the targets of Tirzepatide and Retatrutide that we've shown, GIPR and GCGR. And we're looking here at bias between arrestin and CAMP signaling. On the left are, again, our bias graphs that are normalized to the maximum signal in each assay. And we're using our neutral agonist GIP for the GIPR assay and glucagon for the GCGR assay. Moving to the right side of the slide, the bias plot demonstrates that neither compound is biased to the endogenous ligands for either receptor, either Tirzepatide or Retatrutide for GIPR or Retatrutide for GCGR alone. Interestingly, we do see that all 3, including the natural agonist GIP, have a slight intrinsic efficacy imbalance toward the GS pathway for GIPR, which you can see in this plot down here on the right, being just above 0.5. Now, prior to moving a program from in vitro profiling to in vivo experiments, it's important to confirm that the activity that we've seen against the human receptors is consistent with orthologous species, such as mouse or rat, where some of these in vivo experiments will ultimately be performed. On the slide here, we're showing beta-arrestin recruitment data with these mouse receptors compared to the human arrestin assay. They have comparable potencies and efficacies. This is somewhat expected. The mouse, GLP-1R, GIPR, and GCGR receptors are all well conserved and share between 73 and 92% homology with their orthologous human receptors. In addition to these arrestin assays, we do have second messenger assays that we can be performed with each of these orthologous targets. And for some receptors, we even have rat assays as well. Additionally, we have custom assay capabilities that can be used to develop new assay targets or readouts for anything that's needed for a given discovery program. In addition to all the targets and assays that we've shown in our workflow today, Eurofins Discovery also offers a large range of assays and formats for receptors that are associated with obesity metabolism off the shelf. On the chart here, you can see quite a few more of these. They include serotonin receptors such as 5-HT2C, a broader family of calcitonin and amylin receptors, where we have assays with each of the different ramp accessory proteins. And we even have non-GPCRs like the TNF-alpha family of receptors. In addition to the cell-based formats we discussed in the talk, we also offer radioligand binding assays for a variety of human and mouse targets, and have custom assay capabilities to develop new assays for targets or readouts that are needed. Finally, as you can see on the far right side of this slide, these cell lines can be purchased as products to support for your in-house discovery efforts. The last step in our workflow includes preclinical studies, such as safety pharmacology, pharmacokinetics and pharmacodynamics, and in vivo testing. Once a lead compound is identified, it's important to perform additional preclinical studies, including safety pharmacology profiling, to de-risk compounds of potential adverse drug reactions prior to any further efforts. Here on the slide, we have data for Semaglutide, Tirzepatide, and Retatrutide in the Safety Scan 47 panel. This includes assays for 47 targets that are well established as contributors to ADRs, including GPCRs, nuclear hormone receptors, enzymes, kinases, ion channels such as the hERG channel, which de-risk compounds against cardiac toxicity and is something that's required for all new drugs, and transporters, which include NET and SERT. Data is displayed here for single 1 micromolar concentration of each peptide for clarity. And as expected, we see very minimal response from any of these compounds against any of these targets. I'll additionally note here that the safety panel is run as a full dose response curve that gives potency determinations directly. To demonstrate a specific example of the utility of the Safety Screen 47 panel, we have testing here with a drug called SGI 1776. This is a PIM kinase inhibitor that had development discontinued in a phase 1 trial back in 2010 due to the presentation of cardiac liabilities at the concentrations tested. Testing in the safety panel here shows that we have activity against a variety of these potential risk factors, and this includes the hERG channel that we talked about, which may have allowed them to see this risk prior to taking the drug to clinic. De-risking compounds with in vitro safety pharmacology can save wasted time and money on development of compounds with adverse properties and allow programs to make decisions much earlier on. Finally, the workflow we described today can be expanded to include additional assays and services that we have not discussed. Eurofins has chemistry capabilities for various modalities, and client or Eurofins provided HTS libraries can be used for large screening campaigns to identify hits in any of the assays that we've discussed today. This includes both functional and potency binding assays, and we offer a variety of additional chemistry and in vitro and in vivo, assays to support preclinical validations. Finally, again, cell lines discussed are offered as products directly for in-house discovery efforts. And with that, I'll conclude today's talk by sharing resources that are available at eurofinsdiscovery.com. We have dedicated web pages for the topics discussed, including gpcrBIAS and the obesityLITE panel. You can find linked flyers and white papers on allosteric modulation and GPCR pharmacology, as well as a more detailed resource guide for the obesity services discussed today, and broader information on translational and in vivo assays. Finally, we have a poster that discusses the majority of the data from today's talk, which will also be available as a handout today. With that, I would like to thank you all for your time and attention, and we would be happy to answer any questions that you might have. Thanks.
Thank you, Luke, for your excellent presentation. We now have 3 polls for our audience to participate in. If you could take a moment to click on these, we would greatly appreciate it. Also, as mentioned, we will be sharing a handout. Please ensure you click and download this before the webinar ends. We now have the pleasure to introduce our questions and answer session. Don't forget you can still submit your questions using the questions panel in the menu on the right. So let's take a look at our first question. Given these therapies are injectable peptides, are there other aspects that should be considered during testing at this stage, such as injection site reactions? Luke will answer this question after the 2 remaining polls have been answered. Okay, so moving on and returning to the question, for Luke, your first one is, given these therapies are injectable peptides, are there other aspects that should be considered during testing at this stage, such as injection site reactions?
Yes, thanks, Ellen. That's a good question. So MRGPRX2 is a peptide receptor that is agonism at this receptor is indicative of a lot of injection site reactions and anaphylaxis. So we do offer MRGPRX2 assays, both in arrestin and second messenger format. And we have human and a couple other species of orthologous assays as well, including canine, porcine, and NHPs.
Do you have ortholog assays for rodent species?
Yeah, again, so we had on that chart in toward the end of the slides, we had a assays for mouse and rat for quite a few of these targets. Again, like I just said, for MRGPRX2, we have some other species as well, and we also have custom capabilities where we can develop assays for orthologous species for any targets that we might be missing at the moment.
Fantastic, thank you. Have you tested other modalities like small molecules, antibodies in these assays?
So we have, in our EFC technology, we've used all sorts of different modalities, small molecules, antibodies, peptides, large biologics, antibody drug conjugates. Specifically in the panel, we've also tested a small molecule in our obesityLITE panel, but we can pretty much deal with any of these modalities as needed.
Can you add other assay targets to the existing obesityLITE panel?
So the panel itself is a set 25 assays that we showed here, but all the other targets that we have can be added as an ad hoc assay in addition to anything anybody wants to run with the panel. And we can also work with customers for custom panels as needed if there are other targets that anybody wants to run as a regular submission or anything like that.
What are the different pharmacological modes captured within these assays, such as detecting allosteric modulators?
Yeah, so we discussed agonism and antagonism for the most part in this talk, but we do have the capabilities to detect allosteric modulators, either positive or negative allosteric modulators in pretty much any of our assays. I think on the last slide there, we have a resource page related to allosteric modulation that people can find on the website. And if anyone is interested specifically in that, we can send them that information.
Fantastic. Please join me in thanking Luke. As you leave the webinar today, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.